Everybody, welcome to our webinar. So our speaker today is Aldo Guzmán Sáenz. I hope I pronounced it correctly. So welcome, Aldo. He's going to talk to us about applications of topological data analysis to computational genomics. So please, Aldo, the stage is yours.
Thank you. No, the pronunciation was perfect. Right. So I'll share with you a couple of applications of this TDA to the area I've been working on for the past 5 years, which is computational genomics. I'm currently part of the computational biology group at IBM Research. So the two applications I will mention will be one to signal enrichment in metagenomics, and the other one to phenotype prediction from genotype data. And of course, I will go into what does this mean at all, right? First, maybe a little bit of an introduction, but I'm sure everyone here is familiar with persistence or persistent homology, but just to remind everyone, or in case someone is not very familiar, we can think about persistence in a way that we can think about persistence like a way to keep track of features in something, right? So, for instance, here on this diagram, I have on the left something that could be like a sequence of topological spaces, right? And then to the right, we have something that could be, for instance, their homology, connected by the homology, the morphisms induced by the homology functor, following the filtration. And we have for, in particular for homology and nice filtrations, we have this decomposition theorem that provides us with a way to very concisely explain summarize what features are important in this filtered topological space. So in the diagram itself, I mentioned directed category, but that's just a generalization of the directed sets that have, which are just sets that have a preorder with upper bounds for all pairs. And maybe the main idea is that it has a direction, right? So this sequence idea is that it has a direction, and we want to measure the changes along that direction. Sometimes we don't even need something like a filtered topological spaces. We only keep indices to work with. Sometimes we also don't have filtered topological spaces. We have filtered simplicial complexes, which are the ones that we actually use to perform computations, like in the computer. And there is a nice theorem that tells us, that gives us good guarantees about the information we are getting. So for instance, in this slide, you can see what I mean by this tracking of features, right? This is just a simple example. We show a point cloud, right? And we grow disks around each one of the points in the point cloud. And when two of these disks meet, we add an edge. And when three of them meet, we add a triangle. And of course, we could, if four of them meet, add a tetrahedron and so on. But in this case, we're only showing the two-dimensional skeleton of the simplicial complex. The check complex does recover the homotopy type of the union of the disks, but in practice, more often than not, what we use is a filtered Vietoris-Rips complex that approximates to some extent check complexes. So we don't really lose like a lot if we consider Getter-Rips complexes. And that's pretty much the idea to keep in mind if persistent homology is kind of new to you. You are tracking the features that appear. And so in this case, we see that we have these 3 cycles that correspond to these 3 lines, right? Because this is in degree 1 homology, and in degree 0, we have that each point starts as its own component, connected component, but at some point, all of them kind of join together in a single connected component. And there are some nice artifacts like these short bars that correspond to maybe some cycles that appear for a very brief moment and very quickly get filled out. Are there questions so far? Okay. So moving on to the first application, this is signal enrichment in metagenomes. Oh, I'm sorry, my computer just updated. Hopefully you still see everything. So this is work we did with Nina Haiminen, Shogata Basu, and Lakshmi Parida. Nina and Lakshmi are colleagues at IBM, and Shogata is a professor at Purdue University. So first, let's talk about a little bit of nomenclature. So here we have a cartoon, right? This is only schematic, and we can imagine that we have a collection of microorganisms. They might be bacteria, and these can be recovered from a sample that taken from soil, food, or other possible sources. And you can, and the main thing is that you want to have multiple organisms present in that sample. And what happens is that this is like the genome of each one of these organisms, and you sort of break it up into short sequences. That's just kind of the way sometimes the information about the genome of these organisms is recovered, and metagenome is kind of like all this information about all these organisms at the same time. Okay, so it's in that sense it's kind of beyond the genome, right? Because it's like a bunch of genomes all together. But since they are kind of broken up, they even though sometimes you can assemble them back together to form like a larger piece of DNA or even the complete genome, sometimes you either don't want to or you cannot. So you might want to do something about that. And the problem is then to say, okay, I have all of these short sequences of DNA, which are called reads, like here, reads. And given a bunch of these guys, I want to say what organisms are present in my sample. So there are databases that are, that researchers have put together where you can query sequences, these reads, and ask what organisms have those reads in their genome. And so you could have a situation where, you have a lot of unique reads mapped to a single organism. And in that case, you can be sort of convinced that that organism is actually there, right? Because there are sequences, there are reads that map to this organism, but no other. So the only explanation other than some form of contamination or defects is that the organism is present. But this is not necessarily always the case. What happens is that you get a lot of reads hidden a lot of organisms at the same time. So you want to ascertain which organisms are actually present in the sample, right? And it turns out that just by counting reads and tallying, how many reads a given organism is being hit by is not enough. It's actually, it can lead to this problem of getting false positives. In here, a false positive means that you are saying that an organism is present in the in the sample when it's not. So we want, true positives. And in this particular work that we did, the task was to to determine which geotypes of bacteria were present for a given species of bacteria. So before, when I mentioned that the organisms could be of the same species, then this can actually happen sometimes. Or it could be different strains or something, or things like this. Okay, so we're going to focus then on situations where these uniquely mapped reads are few. So we're going to pose the problem. We're actually going to do it in two ways. This is the first one. And what we do is we, just name the organisms from X1 to Xn. These are not anything, not like sets or anything. This is just the name of the organism. And we are going to denote by Sy the set of reads mapping exactly to that particular set of organisms that we're saying that Y is. Okay, so Y is a subset of the set of all the organisms, and Sy are the reads that we have that map exactly to those. And so the input that we're taking for our problem are the reads, the organisms, and the relations among reads and organisms. Okay? And we are going to take each one of these k-tuples as a vertex of our simplicial complex. So what we want to do is then filter our simplicial complex in such a way that we will get, hopefully, some information about what organisms are present. And this is why we chose this particular filtering function. We denote it by d of y, which sort of intuitively, it kind of denotes how many reads are stacked atop a particular portion of a genome of an organism. So for instance, let's say that you have a bunch of reads in Sy, right? And Xj is one of the organisms that is hit by these reads, so it's in Sy. Maybe let's think about this as being in Sy, for instance. And if you imagine a bunch of reads stacked very high here, this number, right? This length is not changing, but this number is increasing, right? So the depth increases. So this is why this guy is the depth. That's kind of like the intuition behind it. And this will act as a filtering function in reverse, actually. And in here we have this kind of normalization factor because we need to account for the number of organisms that we're currently looking at in this case. So this is the actual, construction. What we do is we start adding. We start with a simplex with vertices, the organisms. So, for instance, if we had 3 organisms, this would be our simplex, and we subdivide it. We take the barycentric subdivision of it, and within this barycentric subdivision, we're going to take this subcomplex that is given in this way. So we're going to take all of these collections of subsets, such that this thing is non-empty, and this thing means only that the depth of the organisms in all of these subsets of organisms has exceeded, a given threshold. It's kind of an involved definition. So for now, maybe just let's think about this as, as the depth increases, you are going to see more stuff. So 0 depth means, for instance, actually I'm going to go back, sorry about that. If we don't have many reads, right, this number becomes kind of slow. It's small, sorry. And this kind of thing, is the one that is going to keep track of the evolution of the filtration. So there are other things to note here. So, for instance, once we compute the persistent homology of the simplicial complex that we get there, we want to then map each one of the generators in homology, first of all, to a representative of this class, right? And then somehow map that to a single organism. So this is already, already going from cycle representatives to just going from cycles to cycle representatives is already, ambiguous. And this has now become harder because we're going from the representative to organisms themselves. So what we ended up doing was, okay, in this case, we have a lot of control over all filtration. So we can actually, decide what representatives are actually useful in our case. And then we took for a given organism, we looked at where it was the most represented, in that when it was more present in these Y's that make the simplices. So, for instance, if you take a given organism and you see that it appears a lot in a certain cycle, then you say, oh, okay, so I'm going to associate this cycle to this organism. And then we created a voting function by aggregating the bars in a way that I am not showing because it's just another formula, but it has to do with the length of the bars and where they started and their dimension. So, for instance, bars at dimension 0 don't have the same weight as bars of dimension 1. And then we add all of this together to get, for a given organism, a value such that the higher this value is, the more likely this thing is telling us that this organism is present in the sample, given all the reads that we are considering. So in these results, this is simulated. These are simulated shotgun reads from 36 organisms. Shotgun reads means pretty much the idea that I mentioned before, that you have like a bunch of reads like that are sort of subsampled from these genomes, and ideally you would like everything to be concentrated close to the upper left corner. And we see that when we have one true positive, this method gets it, and where are two true positives, it sort of gets it. We would like it to be in one as well, because you want the voting function to tell you, okay, one means it's a true positive, and the lower you go, the less likely it is to be a true positive. So it kind of gets some information, but not everything. And similarly, in this case, with the positives, it also it kind of goes down by a lot in some cases. So it seems that this method does miss and some true positives, in that they rank lower than some false positives. But still, this performance is not that bad, and it's, when compared to naive counting, it doesn't, it's not that terrible or anything like that. But since we observed that it wasn't, these results are not like the best we could do, we said, okay, let's think about it another way. So next, we considered just taking common reads to simplices. In this case, k-way common reads means that you have reads that are shared by k organisms. Right. And so we filter by how many common reads are there in each one of these simplices. And then we again go to the barcode, and we do this mapping back from bars to representatives to organisms, and we get from that our voting function. And here's like a word sample of what we mean here. We can think about it in that we have 4 organisms, right? And we can ask how many organisms are present in both, for instance, in A and B, or in B and C, and so on until in everything. And the cardinality of the size of the tuple that we are taking, right, is the dimension of the simplex, right? And the number of reads that we're taking is the filtration value. It reversed, of course. Again, it goes backwards. So after doing these constructions, we did get improved results over the previous method. And so we were kind of happy about that. I mean, it would have been nice to have both of them do well, but it is what it is. And these something maybe for students out there, it's something to consider, is that sometimes you maybe don't want to consider just straightforward point clouds in RM. Maybe you want to consider things like metric spaces and so on. So yeah, that's kind of like the main point of this particular application. This voting function that tells you how likely a sample it is to be, how likely an organism is to be present in a sample. Yeah. I think I made a mistake explaining the depth actually, but I think it's maybe a little too late to go back to that. So maybe if there's time at the end, we can look at it again. Are there any questions here? I don't see anyone, so I'm going to take the silence as a no questions.
No questions in the
I'm
chat,
sorry.
so I guess we're fine. Yes?
Oh, so,
Hi, this is Radmila. I would actually like to ask for your insights, maybe about the interpretation of the one-dimensional cycles. If you have anything else that you have observed, because this is a quite involved process. So you're combining sort of the different dimensions. So what's the motivation behind it? And so we all know how to interpret like the zero dimension, but what about the first one?
right. So for instance, you can think about, in this case, for these constructions, we're asking about the presence of a cycle, right? So the cycle in this case would mean that you have reads that are pairwise present, but not triple-wise present, right? So there are some reads that are unique to one of these three guys, right? But not the other two. Otherwise, you would have filled your triangle. So we want to keep track of how far these cycles go along the filtration until they get filled in. Right, and so the interpretation is that these three organisms are related, but not that related, kind of. And that's why we take it into account what's the name? The voting function.
That's right, and that's why you do sort of like the check versus Viatori strips thing. Okay, thank you.
Yeah.
Thank you so much.
Okay. So now for the second application, we're going to see about predicting phenotypes from gene expression data. And this is work with Shayan Mandal, Nina Haimenen, Shogata Basu, and Lakshmi Parida. Nina and Lakshmi, I have mentioned that they are colleagues at IBM, and Shogata is a professor at Purdue, and Sayan at the time was an intern at IBM, and he was in Ohio State University. Now, I'm not sure where he is. Sorry about that. Okay, so first, again, let's talk about the nomenclature, right? So what is gene expression? Well, when people talk about gene expression, they tend to mean the two steps that are related. Like this white disk on the left, we can think about it as a nucleus in a cell, in a eukaryotic cell. And what is happening in that disk, in the nucleus, is that the DNA is being processed to generate something called messenger RNA, which is this mRNA. And the process of taking the DNA, sort of unzipping it like this, and then generating this mRNA is called transcription. And there are some like, all of this is kind of like molecular machinery. So there are all sorts of things happening here. But the thing is that you go along the DNA, you unzip some parts of it, then you take advantage of the fact that some biochemistry connections are complementary, and then you generate the mRNA. As you can probably guess, I'm not a biochemist, so I will not go into how this exactly happens, but the bottom line is that something goes along the DNA, it generates something that carries the information of that particular segment. This is why this is like a message in that sense. Then this guy travels outside the nucleus and is now here, and it gets processed by something called a ribosome. And inside the ribosome, the process that happens is that another RNA comes in. This one is called transfer RNA that has a certain shape in here that matches the shape that it's being attached to, and it carries an amino acid. So when these tRNA, several of them go in and then go out, go in and then go out, and what you end up is with a chain. So the first tRNA goes in, you output this guy. The second one goes in, you output this guy. And the third one goes in, and this guy comes out. And this is a polypeptide chain, or a protein. And I will say this is like really fascinating stuff. And this step is called translation. So on the left, we have transcription that takes DNA and produces mRNA. And on the right, we have translation that turns this mRNA, or not turns it, but with the help of the information in the mRNA, produces or synthesizes protein. Okay. So this is of course very simplified, but this is roughly what transcription and translation is. And then we say that a gene is expressed if this kind of thing happens a lot. If we see a lot of the products that come out of a given gene, and just to kind of say what a gene is, because I haven't said it right. A gene, we can think about it just as a portion of the genome in the cell. So if we imagine this DNA strand being very, 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 very long, this helix very long, a gene is just one portion of this helix. And a gene is just one portion of this helix, and it can be contiguous, but it might not be. So, for now, let's just think about it that way. And that's kind of what gene expression is. You see, it's how much a gene is being expressed at a given time or circumstance, and that means that you are seeing a lot of the product that is expected from the gene, which could be something like protein, for instance. Right. So, for instance, a natural question is, well, okay, but then how do you even see if a gene is expressed? Well, we can see it like in the cells, but we can also see it in our cells, like with our actual eyes. For instance, if someone has a certain trait that is known to be associated with a certain gene, we can also say that that gene has been expressed. But in this case, what we would like to know is what the levels of the gene expression are. So we want to actually quantify this instead of just sort of qualitatively, look at someone and say, oh, okay, it seems that gene X is, expressed on this person, or it got activated or something. So what happens is that people in the lab, right? Researchers, they sometimes quantify the presence of the proteins that we saw in the diagram that is in the previous slide, or they see or they check the presence of the actual mRNA transcripts that are present. So that's and this is a very involved and complicated process, and it's it's challenging, I guess, for for people to come up with these new ideas to do it even better all the time. And there's been a lot of progress. But yeah, these are like two possibilities: either see how much protein you have, or see how much mRNA of that particular, associated to that particular gene is present. Okay, but after all of that happens, we can then say, okay, just what I'm going to work with. Well, after all this counting and quantifying of the product of associated to that gene, we get a number, a non-negative number that tells us how much or how high the level of a gene expression we have. And this is kind of the input that we are going to work with for this particular application. But I just wanted to give you like the context, from where these numbers come from, because for me at least, at first it was kind of mysterious, like what's gene expression levels, what's okay. They are numbers, but where do they come from? And I wanted to give a little bit of background on that. Okay, so we get these numbers in this matrix as input, and we want to see what happens with a phenotype depending on these values. And the phenotype we worked with in this paper was Parkinson's disease. And maybe you are familiar with Parkinson's disease, but just in case you're not, Parkinson's disease is a neurodegenerative disorder that affects many things, but in particular, it affects motor functions, and also there are some non-motor symptoms like cognitive impairments and disorders in sleep. So there's a lot of research into what causes Parkinson's disease and how to deal with it. Okay. And so we said, okay, so let's give TDA a shot at the task of predicting this phenotype from these gene expression values. And let's see what happens if we take standard machine learning methods, like off the shelf, and if we don't do fancy things like grid search and hyperparameter tuning and so on. Let's see what happens with both of these approaches. And just to give you a sense of the size of the data we worked with, for this particular work, we used about 20,000 genes and about 400 subjects. So we can imagine that we have a matrix of 400 by 20,000. Okay. So the idea that we were working with was to have a sort of topological signature for each patient, right? And we wanted to make it so that we would be able to use a workflow like in machine learning, which would be, you train first, and then you predict on seeing data, right? And there could be a way to do it with, for instance, you could generate a point cloud given each one of these rows, right? By sliding a window, right? So you do this kind of mapping thing. If we knew that there was some sort of spatial coherence or something across the genes, right? Because then you say, okay, so gene 1, gene 2, gene 3, this is these 3 coordinates, right? I'm mapping them to R3. So I put that point there, then I take gene 2, gene 3, gene 4, and so on. But in this case, it's not clear, if this spatial coherence is available, because some of these genes sort of overlap, or they might not be contiguous, and so on. So we thought, well, we could instead see how much a gene is related to another gene in a subset of this subject set. So we would measure something like a correlation between them, and then use that as a sort of notion of distance between a couple of genes. And the thing is that we actually used Peterson's correlation. Of course, we transformed it so that it would become a distance. But we didn't see very good results there. So then what we thought of using was distance correlations, which has slightly, different information that Pearson's correlation, in that, measures nonlinear associations, and it has some good properties that, it's good to have. Okay. So this would give us, if we apply it to here, then we would get a bunch of points in space. Of course, here I'm putting it in here, but in principle, it might not even be like embeddable, right? It might not be like something nice that we can visualize, right? So just to give some background on what distance correlations are, this is kind of how you define distance covariance, and you take this kind of norm of these characteristic functions of x and y and their joint characteristic function, and you take this norm. Now, this norm is it's not exactly, like easy. You have to add a sort of scaling term in the integrand. And if you want to see the full formula, you can look at this paper. But once you have this guy, then you can define this distance correlation. Square in this case. And this gives us, these nice properties, and it has guarantees about independence of variables when something is 0, and many desirable properties. So this is what we use to measure how far or close to genes are in our training data. I'm going to say training data now to distinguish from the training to the testing data, which is data that was not seen by any of these constructions. Okay. So in our case, we ended up with a semi-metric space. It failed to have no, I think this is a typo. Yeah. Well, okay. So we ended up with this space and this notion of closeness. And the idea we had then was, okay, so we had this point cloud, but this point cloud, I mean, that's good and all, but what we wanted was a topological signature for each one of the subjects. And so what we did was we augmented this point cloud with weights at each point. The way we use the weights, or what the weight was, I should say, was depending on how much that gene was expressed in that particular subject, right? So a gene that wasn't expressed a lot, has a small weight, that kind of thing. And so we then used, we constructed a weighted Dietrich complex, right? And we had to actually solve some, identities, and we ended up with that particular formula that tells us when to add each of the simplices. And here on the right, I'm showing what it means to have these weighted point clouds, right? So for instance, one point cloud could look like this, and another one with different weights could look like this, right? And the thing is that this gives us different simplicial complexes for different subjects, and so now we can think about computing persistent homology, for instance, right? And here we run into more issues, which were, in particular, that there were like a lot of points in this point cloud. So, this weighted Dietrich thing was a little bit problematic. So what we thought of doing was we started subsampling and averaging. But of course, we cannot just, take the barcodes and do it with them. So we took persistent landscapes, and we began, this process of subsampling and averaging. And then once we, for each one of the subjects, once we got their average, this kind of subsample average of for each one of them, we vectorize them. We vectorize them, and we did it in this fashion. We just, quantize everything, right? So quantize means, sample some finite set of points in both, domain and however the lambdas from the persistent landscapes are present. So the lambdas themselves, since they are lambda 1, lambda 2, lambda 3, and that's kind of already discrete. But on the filter function, that's where we kind of had to sample and give a sort of a resolution there. And then we thought about feeding this into ML classifiers, right? But then we thought, oh, okay, well, we're getting these landscapes in different degrees of homology, so why not put them all together and just use neural networks with them? You have this kind of looks like an image, right? And this guy looks like an image as well, so maybe we can put them together or something and see if you're looking at a single image but with two different channels and that kind of thing. Well, because we were working with zeroth and first degree homology. So that's what we did. We started working with convolutional neural networks. Maybe some of you are familiar with them. Here I'm showing how they work like in a one-dimensional example. So what you have is a filter, which is the red block thing, that thrives, across what you get from a previous layer in a neural network, and then it produces an output that goes, that is the, the result for the next layer. In 2 dimensions, you can imagine this guy being like maybe 6 blocks, like 3 on the left, and like it has another part here, and this guy as well, and it now travels in 2 directions. Maybe it first goes rows and then columns. It's depending on the implementation. But the idea is that you have this kind of coherence, right? So it has this kind of, each one of these filters looks at, in this case, at 3 things that are close together at once. Okay. And so multiple filters will give us multiple channels, and already, convolutional neural networks have proven themselves to be very, very good, for visual tasks, and they have enjoyed a lot of success there. So we thought to use them for this classification task. So, okay. So now let's summarize because it's kind of a lot. So just to summarize, we began with our matrix, right, with our table of subjects and genes. We took some subset of rows, which was our training data. In that, and with that training data, we computed this point cloud with vertices genes, and then for each one of the subjects, we computed a weighted point cloud, right? And from that, we computed a weighted Dieterich-Rips complex. And from that, we got this persistence landscapes. And that's what we're going to feed into this convolutional neural network. And the convolutional neural network, the task it is going to solve is to classify. So we know the labels. We know, people, subjects that have phenotype Parkinson's disease, and we know the subjects that have phenotype not Parkinson's disease. And so we train the neural network on this data, and then we test it on the unseen subjects. Now, we can actually use this point cloud, this gene point cloud that we got from training data only with the test data as well, because the way to construct the weighted versions of the point cloud and the Dieterich-Rips complex is by looking at how high gene expression levels of a given gene were in that particular sample. So we can use the training information with the test information. So in this sense, we have achieved what we wanted, meaning that we got something out from the training data that can be now used with unseen new data. Okay. So we did this prediction task with the testing data, and then of course this has to be done with cross validation, and you have to split your data several times, and look at the average or the distribution of the scores and so on. There was a very slight class imbalance in input data. It didn't really make a difference. And so what we compared against was with SVM with a linear kernel and SVM with a radial basis function kernel, with random forests, and with a fully connected neural network with several layers. In this case, it was, an MLP, right? So what we ended up getting was that somehow this pre-processing of data with TDA made it so that the convolutional neural network got very, decent results. Like if we look at the true positive rate and the true negative rate, right? We see that they are both high, and this is kind of important because sometimes what you observe is you get one that is high but the other is low, meaning that your classifier is kind of predicting something being in one class, and just so you said that almost everything was in that class, so you got almost everything right. So we see this kind of imbalance in these guys, and therefore their scores solver for this is the F1 score. So all in all, you know, this preprocessing of data helped a lot. Although I should mention again that these other classifiers were used with minimal fine-tuning. There was some fine-tuning to improve the results, but there was no like additional preprocessing on the data there, other than like the standardization of variables and so on. Oh, right. And the input to these guys was the actual matrix, right? So that's another difference. Right. So all in all, you know, we got these features and it was okay. But then an interesting question is what are the features that the neural network is looking at that, you know, make it easy for it to distinguish these classes, right? And that's only half the problem. The other problem is, okay, well, once you figure that one out, well, you know, what this persistence landscape is looking at, you know. So those are, I think, non-trivial questions. And a natural question to ask is, well, you know, this time we chose distance correlation, but what happens if you do something even fancier, right? So that's something we might try in the future to see if it improves or not the performance of the classification task. There has been work in saliency, feature saliency, for this kind of thing. And with respect to these 2 points, there's been some work about choosing, like, for more general instances, how to choose representative classes for a given bar, but we haven't been able to work on it, apply to this setting yet. So that's something we might look at in the future. Oh, right. So we use our in-house solution for this, and I have a slide here for some reason about it. It's meant to be like a toolkit. Yeah. So I think that's it. Thank you.
Thanks very much for this very interesting talk. Very nice. So I would ask you for questions. Do we have any questions for Aldo? Please.
Hello, just for curiosity, for doing all this, do you need really a large, a very strong computational power?
Right. So yes. For instance, in the simulated, in the first application, in the signal enrichment situation, we got in that case, we didn't really need a lot of computing power because our simulation was small, but it very quickly can grow. So then you would need, a lot more. But in the second one, you do need a lot of power to you know, both construct the complexes and compute their persistence. The classification task is actually not that difficult because we're even with a very small convolutional neural network, we got good results. But the TDA aspect of it, that took some some non-trivial computing power. Yeah.
more questions, please? So I was
Yes.
wondering, your training sets, how big were they? Your data sets?
Right. So let me see if I can just jump to the slide. Right. So for this particular work, we were working with 400 in total, right? About 400, and we did splits of 80-20, 80 training, 20 testing.
So comparable to other. Sorry, go ahead.
Oh, I was just going to say that they were stratified splits. Yeah.
So I was going to ask, compared to other comparable researches in this area, was this a big or a small data set?
This I wouldn't say it's big. Yeah, I mean, right, because you sometimes have thousands of subjects.
Mm-hmm.
Right. But I guess what made it difficult in this case was that since we were working with the transpose, we had suddenly thousands of points in our point cloud, and we had to subsample. Yeah. But it's not unusual to see, oh, and in this case, since we were working with genes, there is like a sort of quote unquote small upper bound on the number of genes that we have for humans, at least. But if you're working with other kinds of data that look at the genome in more detail, you can have a lot more features, right? So you can have like millions, or
Mm-hmm.
Yeah.
And of course, that contributes to the complexity of computations, right? The TDA part.
Right, exactly.
Mm-hmm.
So it would be good to have something that helps deal with that kind of thing. Yeah.
Any more questions, please? Well, if not, thank you very much again, Aldo, for this very interesting talk. And thanks everyone for attending, and see you next time.